This conference will now be recorded.
Welcome everybody to today's GEO AquaWatch webinar. This is the second in our forecasting webinar series. My name is Dr. Mary Beth Neely. I'm the scientific coordinator for GEO AquaWatch. I am stepping in to do the introduction today for our director, Gada El Sarafe, who is on bereavement leave right now. We have three speakers today who are going to tell us a little bit about the state of the science and forecasting from their diverse perspectives. Our first speaker will be Dr. Rick Stumpf from NOAA. He will be speaking on HAB forecasting. Rick is a member of the National Centers for Coastal Ocean Sciences. He's an oceanographer there. He has over 40 years of experience in satellite remote sensing as well as modeling research. Our second speaker will be Dr. Ralph Quast. He's with Brockman Consult in Germany, and he will be speaking on the forecasting of water quality from space. Ralph is a researcher and software development expert in Earth observation. Again, he's in private industry with Brockman Consult. Our final presentation will be co-presented by MVIS's Evangelos Romas and Apostolos-Tzimas. Evangelos is a hydraulics and hydroecological modeling researcher with MVIS, that's a consulting company, I believe based in Greece. And Apostolos is the managing director of MVIS, working on lots of European Union projects, including Prime Water, and that will be the basis for their presentation today. A lot of the forecasting recommendation outcomes from Prime Water. Our first speaker, Rick, you can go ahead and take over the presentation.
Okay, thank you, Mary Beth. Good to be here. I'm going to talk about work we're doing on cyanobacteria blooms. This case, I think most everyone here would be familiar with those, but one of their conditions is producing dense scum, sometimes on the surface of the water, although not always. I'll just go into here. Let's see, started. They're a widespread problem around the world, but focusing on the U.S. in this case. And I would say that there's risks as far as managing drinking water, both the biomass as well. Recreation is also an issue for pets in the United States. There's always, unfortunately, several dogs each summer that die after swimming in lakes that have cyanobacteria blooms, toxic blooms. There's also aesthetic point of view, and some of these even come into estuarine and coastal waters when they're influenced, when they're near some of these strong bloom areas. I think someone might need to be muted. Okay. So there's within the U.S. there's cyanobacteria blooms all around the United States, and you can see here every state gets affected by it. In addition, while they're not always toxic, but there are toxins present in all of them. Anything where there's a color on this map, toxins were detected at some level. And the WHO one, I should note, that's 10 micrograms per liter. That's a little higher than the U.S. recreational recommendation of 6 micrograms per liter. So there's actually many of those blues are a significant risk. We've been working with Maris and Olchi for detection. I won't go into the methods, but we have several papers on this. taking advantage of the wiggles in the spectra and that pick up phycocyanin and chlorophyll A. And we draw on, essentially the algorithm, we draw on phycocyanin for detection and chlorophyll A for biomass. People might ask, why not use the phycocyanin? And a couple of issues. As a practical one, almost no one measures phycocyanin. And at least in the United States, there's no standard methodology for measuring it as well. And there are challenges with that. The second one is the blooms themselves produce and phycocyanin as they need it. And so phycocyanin concentrations per cell vary considerably within cells. So you can have a difficult time trying to sort out how much bloom you actually have based on that. So it's an excellent indicator for freshwater blooms. It's not for marine blooms. If people work with trichodesmium, they need to be looking at phycoerythrin or other pigments. But chlorophyll A gives the indicator of the biomass once you know bloom's present. And this is an example of Maris image of one of the severe blooms in Lake Erie and the Great Lakes. And just some examples of some other areas. This is over this section here is Lake Winnebago, Green Bay, that's Lake Michigan and Wisconsin. Down in Florida, there's actually, this is Lake Okeechobee, quite large, it has significant blooms, but there's actually cyanoblooms in a number of these other lakes. And several of these actually, there's no scum present when these blooms occur. So that's important to keep in mind. We're not looking for scum in these areas. How well does the method work? We established a relationship with cell counts in Lake Erie and then tested it at a number of other places and found that that relationship of the index to cell counts held up very well going across these regions. And then we similarly did this looking at the chlorophyll around the country and established a relationship to estimate chlorophyll as well with this. So when we do chlorophyll, it is chlorophyll related to cyanobacteria as compared to all chlorophyll in the water. That gets to a whole other topics there. So it's effective in that means. In Lake Erie, one of the blooms actually led to a problem in drinking water in Toledo in 2014. There was a breakthrough of toxin from a bloom that had started earlier than usual, more intense than usual when it started, and was producing far more toxin than normally anticipated. We'd had, in 2014, we were doing about weekly monitoring with MODIS data, which is somewhat problematic at the time, but as Maris had failed or had been lost, and there weren't other monitoring mechanisms. So they were surprised at the time. And a key issue was to establish a system that provided more rapid and better information on when the blooms would show up and when they would show up near drinking water intakes. Because it's not enough to know where they were, say, yesterday or the last clear image, but it's also important to know where they're going to be coming forward. So we've looked at these from two sides. One is doing seasonal forecasts. How big might the blooms be during the summer? So we can anticipate, so that allows communities, municipalities, governments to plan logistics, supplies for treatment and the like. And then we're also doing short-term forecasts. So cyanobacteria are driven primarily by excessive phosphorus in the lakes. Some lakes already have a load of phosphorus inside. Lake Erie, among many others, their phosphorus comes in from external loading, but phosphorus itself is key to that. The more phosphorus you have, the more likely you are to have cyanobacteria, the more you're going to have. You have a fundamental change. As far as Lake Erie, the type of phosphorus matters. Total phosphorus, which is actually shown here, hasn't changed much. Well, it decreased after the U.S. Clean Water Act was implemented. It slowly came down, and that's a result of controlling sewage and industrial inputs to phosphorus. And so for a while, the blooms disappeared. On the other hand, dissolved reactive phosphorus bottomed out in around late 1980s, early 1990s, and then has been increasing since then, or increased up into the 2000s. During the 2000s, blooms reappeared in Lake Erie, and just about all of the dissolved reactive phosphorus, the DRP, is biologically available. So unlike particulate, most particulate phosphorus these days is bound to sediments. In earlier times, most particulate was in sewage and the like, but now it's bound to particulate, so a lot of that's not necessarily available. So the increase in dissolved reactive phosphorus is a key factor. This is believed to be a result of changes in farming practices. There's a whole variety, tile drainage because of the poor drainage of the land. No-till farming changed how fertilizers were applied, and so it was actually easier for fertilizers to run off as a result. So combinations actually led to these increases. With the monitoring we've been able to put together with Maris, we managed to get a MODIS matched to Maris with a CI equivalent, and then going with Ulchi. This is the peak for each year, and I just noted some major bloom years in red, the red circles, and they can differ quite a lot each year. Going across the lake, by the way, the distance from across here is about 150 miles, 100 to 150 miles that we see here. And those who have not been on the Great Lakes, if you're standing on the U.S. shore of Lake Erie, you cannot see the Canadian shore from it, that giving the size of that. So the blooms vary quite a bit between years. Well, we examined phosphorus load by the main tributaries into the Western Basin. And I should say the primary one is the Maumee River, drains into the southwest corner, and that provides the majority of the flow, about 80% of the flow into that area. The Detroit River is low phosphorus water, and while it's a very high discharge, it doesn't have an influence on this because of the very low phosphorus concentrations going into it. So we focus on the Maumee River load of phosphorus, and there appears to be a relationship between that and the quantity of biomass that forms each year at its peak. And so we then said, if we understand how this works, then we should be able to forecast it. So in 2012, we did our first forecast, and that actually was an excellent test year. We just come off the worst bloom Lake Erie had ever seen and a really wet year in 2011. 2012, interestingly, had a very dry spring, and what we had found, it was not the total load for a previous year, but only the spring load mattered. So we had a wet winter and a dry spring. And the forecast, we'd have a mild bloom, and in fact, the end result was a mild bloom. So our understanding of spring phosphorus load being the key factor driving the bloom was borne out with this forecast. If it had been internal loading or load for, let's say, the previous 6 or 8 months or so, we would have seen a much larger bloom than what was actually observed. So our understanding of the spring load is key to that. We've been monitoring since then. We have forecasts every year, up to 11 years. The 2022 forecast was a little low, but it was in the moderate range, and there was somewhat moderately high bloom. There's other factors going on that changing this, which would require a whole nother talk on what's going on at the lake. And there's indications of climate change actually being a factor, actually shifting these blooms from the first decade of this century to the second decade as well. But that's way beyond what I can talk about today. Just to note that between years, the blooms can vary a lot in size and intensity. Like we don't have an ability right now to forecast well in a month or more in advance how large an area might be covered by the bloom. Right now, it's just the total biomass. We do monitor. Important part of any forecast is monitoring routinely. So we have daily updates. What's great with Olchi now is we're able to turn around the data in the same day. Overpass is between about 11 a.m. and 1100, 1200 hours, and we typically have image posted by 1600 hours, 4 p.m. in the afternoon. So a little over 4 hours after the overpass. And that's quite a statement on EUMETSAT's ability to handle the data. That's where we get it. And EUMETSAT and NESDIS working, NOAA's satellite service working with EUMETSAT to receive the data and get it available to us. So we work on our short-term forecast. We do transport. We use a 3D model to do this. And microcystis is the dominant here, and it moves up and down in the water. It has buoyancy control. Typically is up in the daytime. Once it produces enough carbohydrates, it will tend to sink. Not always, but it tends to sink. And then, so you have this modulation. So an important part then is how much mixing is there, which determines whether it's going to tend to be up at the surface or stay up at the surface. So we examine the mixing and then combine that with a model for sinking for buoyancy, going up and down in the water, and we can run that. Lake Erie has a operational three-dimensional hydrodynamic model based on FECOM, and so we take the output, include the vertical mixing, and add in this biological buoyancy model into it. And so as a result, we're looking in 3 dimensions, and if I can get this to start, okay. So the changes in the concentration, the aerial extent is driven by 2 dimensions, but you'll also see that this is the surface concentration, and the surface concentration is in fact changing as well through the day and also with the wind speed. So winds were very calm, and you saw a lot of orange. Now the wind's picked up. It's about 20 knots, and we've lost some of that surface concentration. So we're able to determine its, look at its extent, and also the likelihood of risk to water plants, which are typically taking water at mid-depth. So they're more concerned about more mixing. And then recreational users, boaters and swimmers, are more concerned about it being at the surface. So we have the ability to actually monitor or provide forecasts that benefit both of those groups by having this 3-dimensional model. And this runs, just let this run a little longer. See some of that transport. Okay. Okay. We've expanded this out with a project called the Cyan, taking the methods we developed in Lake Erie and applying them across the U.S. There's some 1,800 resolvable lakes with Olchi. Those who are interested in this can grab that. We have marks for each one of these. It's a joint effort between several of our agencies now. And this allows us to look at trends in the lakes relative to each other. So some of these, what you see in red, are ones that are getting worse. This is example data from Ohio and Florida, and the greens have actually improved relative to other lakes in the area. That allows potentially managers to start looking at places that have changed and what's going right or what's going wrong. And again, we have a number of lakes across many of the states. And then this gives a sense of the severity, so you, with the data set, can actually look at distributions across the country for it. So we quantify the severity for these blooms as well. And of course, you can actually see a number of lakes in Florida have significant problems, but they're scattered around the country. And there's an ability then to start looking at kind of larger scale factors as far as forecasts, and that would include climatic factors. So we're examining these changes Anything you see in red is either a problem or getting worse. The blue areas are getting better. And interestingly enough, from the Maris time period in the 2008 to 11 versus the Olchi time period, there was actually some improvement in a number of these lakes. And we do have an assumption that if everything gets warmer, everything has to get worse. Well, change is not monotonic, and so we need to be careful about the patterns. Keep in mind, we also have to deal with the nutrient load. So temperature alone, temperature doesn't produce blooms. Temperature helps them grow. And if you don't have the nutrient loads, it doesn't happen. So you need to look at factors like rainfall and so forth. What we have in some of these are indices. This is the Palmer Drought Severity Index, which is basically how much water, and it's a factor in some regions. Days of precipitation above normal are also factors. Temperature does have an influence in the southwestern U.S., some of the desert area, but we're able to start looking at some of those factors on a climatic scale that may be driving changes in blooms between lakes and regions. Just to note, by examining some of these, we're in a better position to, looking at the past, better to identify conditions in the future. In Louisiana, when the Mississippi River floods, there's a spillway just called Bonnie Carey, just north of New Orleans. That water, Mississippi water, is released into Lake Pontchartrain here, which is a brackish water estuary, and that can protect St. New Orleans when this happens. And these blooms can actually come out and end out in what would be standard estuary, higher salinity than they would normally grow, but it's basically been changed into a brackish estuary under these conditions. So we now have a better idea of the potential risk for blooms in these areas, which actually include a number of recreational beaches. We've been working on Lake Okeechobee, which is a drinking water supply and also a source for other places of the lake. And I'll get to a question once I get done here. All right. And so we're now applying a similar approach with a three-dimensional model for going into Lake Okeechobee. This was one developed by the Water Management District. And again, we're using satellite data to initialize the model, as we did in Lake Erie. And we'll be including a three-dimensional transport as well for this. So what's critical is there's an output on the east side and one over on the west side. That water flows in the estuaries, and one of the goals when the water level needs to be managed in this lake, that water is released when the blooms are not present around those outfalls. So that's a key part of this modeling effort to help that management effort, so that they aren't detrimental to the surrounding areas. And with the satellite data, our ability to work on these models, identifying the blooms is critical, characterizing them also, so you have a better sense of when they occur, where they occur. Monitoring is always a form of a nowcast. Blooms don't disappear overnight, and if you know where it was yesterday, you at least have a starting point. But importantly, that can then be used to initialize models to do forecasts. And of course, then the overall satellite data can be used for any statistical model development, looking at seasonal climate change, and also for validation. And I've got a number of collaborators in my group here. I acknowledge, and we get support from a number of agencies and groups. So thank you all there. And I think there was. I saw something. And
well, wonderful, Rick. Yeah, I did post a question for you. You can always count on me for a question, but I will defer to anybody else in the audience who would like to ask their question first. We do have 10 minutes for question and answer.
Hello, Mary. Can you hear me?
Yes, Felipe, go ahead.
Okay, great. So, Rick, and thank you so much for your talk. I'm Felipe from Brazil here. May I give a question? Two questions actually. First, I would like to hear a little bit more about the Sentinel-3 ALSI processing, since it's the level 2, it's not available like for end users easily. So if you don't mind, like sharing some information in terms of pre-processing atmospheric correction of that. And second question is more related to the model, the forecasting model you showed in the last slide. I would like to ask if you have any model available that combines, of course, satellite information with wind and phosphorus concentration and sunlight, all these key parameters for bloom forecasting. Thank you.
For the Sentinel-3 data, we process it using CDAS, although you could use, I guess, Beam from ESA. Actually, we use a mix of those. Our process is minimal. We generate what's called ROS, which is only rarely corrected for atmospheric correction. And that's one optional output. You shouldn't have so it shouldn't be difficult to get the data and the software sources for getting to the reflectances and mapped. They are openly and they're freely available. I can say it's a bit of a learning curve to get them set up. I'm not aware of sources that are already processed. And for cyanobacteria blooms, standard water reflectance is a problem because, for example, if you have scum, you do not retrieve water reflectance, it gets messed out. So going to something that does not have it is better. The algorithm will do okay if you end up with just top of atmosphere reflectance, but there are some other issues with that. On your second question on we've right now for the short term, we're concerned about location of the blooms going out. We go out as far as 5 days, and over that period, growth isn't a primary concern, so we don't try to include a growth model. We have some models that we're working on now that get at seasonal growth, and there's actually a number of what are NPCD model types that are out there that will do the same, and we're looking at different approaches to see about production of blooms. With the satellite data, we haven't looked at actually trying to grow the blooms from the satellite data. That's an interesting question, and we started talking about that, but we're not there yet. So that's another one to take a look at.
We have a question in the chat from Karsten Brockman. He says, can you say a few words about the forecasting methodology that you use?
So for seasonal ones right now, the satellite data is used as the reference for the extent of blooms and the amount of bloom. And we've used statistical model, but we're now moving to a very simple box model to address some of the components there, and that's using phosphorus as an input, also including temperature as to, there's a temperature sensitivity for growth, and so the growth term will be a function of temperature. So simple box model to actually trying to generate a bloom. And for the short-term forecast, we take the satellite data and depending on which software someone uses, we either convert it into particles, move them around, or we move Actually, we do it all as particles. I was going to say concentration fields, but that's a different issue. Because of the buoyancy issue, you really need to keep track of them as particles. So we distribute, randomly distribute particles within all the grids, and then push those around with we take the velocity field, three-dimensional velocity field, include the buoyancy, which right now we have a diurnal buoyancy for the blooms, put those together and run that. So it is decoupled from the model in the sense is that we can use any circulation model to feed it as long as we have a vertical diffusion. So there's probably a lot more you'd want to know. If you're interested, we have papers on each of the methods that we can provide. So get in touch with me.
Great. Thank you very much. I would just point out, if anybody was at our Water Talk, Early Career Science Water Talk, first one yesterday by Annabella Farrell, she mentioned that resuspension of phosphorus from sediments was important. So I wondered if that, that possibility factored in at all to your model.
That depends on where you are. In Lake Okeechobee, phosphorus in the lake is important. It's been so many years of the lake has been there was a lot of very bad management decisions that put a lot of phosphorus in the lake. So there's this layer of muck on the bottom, and that's now a source of phosphorus. So inputs of phosphorus to Lake Okeechobee are actually not so important because it's there. In Lake Erie, the phosphorus resuspension is not a significant factor because most of the phosphorus that ends out on the bottom of the lake is bound to sediments. And in the Western Basin, it's always the water's oxygenated, and if it goes anoxic, you can get phosphorus released from the sediment, but the Western Basin doesn't have that. Central Basin of Lake Erie has a whole nother problem, but so we don't have a phosphorus resuspension. So the answer there is it depends on your water body.
Right.
I can't make an assumption about phosphorus or nitrogen as to what's driving this unless you understand that question. And so some lakes, the bottom's full of phosphorus, and you get a resuspension event, you've just resupplied the phosphorus. And others, it's the input. The only thing that's a consistent thing is cyanobacteria occurs when there's too much phosphorus in the system. There's always you don't lack for nitrogen. You may have so much phosphorus in the system that it looks nitrogen limited, but that's because there's so much phosphorus that you've already got a bad bloom, and more nitrogen just makes a really bad bloom worse.
Great. Thank you very much, especially for answering that. We are going to shift over to Ralph Quast while he's getting ready. I will just point out, Rick, there was maybe one additional question in the chat. If you wanted to respond to that in the chat, we'd appreciate that offline. And Ralph, we're seeing your slides.
Yeah, also, can you hear me and can you see
Yes,
me?
we do
Okay.
hear you perfectly. So go ahead and take it away. Thank you.
Great. So my name is Ralf Quast. I'm a physicist and work for Brockman Consult on a study named Forecasting Water Quality from Space in the role of a data scientist and project manager. The study is funded by ESA, and our work on the study has started recently in September and has a duration of 1 year. Thus, we are yet at the beginning. The theoretical context of the study is time series forecasting. But what exactly is time series forecasting? Imagine that you were given values of one or more variables which have been measured or observed regularly from some instance in the past up to now, like the time series of water quality parameters marked by the blue line here in my schematic plot on the slide. To infer the most probable future values of these parameters, you need to do a time series forecast. Your forecast may comprise merely one time step ahead or multiple time steps ahead. My sketch here shows several time steps ahead. Clearly, water quality parameters are statistically or mostly causally correlated with parameters describing marine physics, atmosphere physics, and marine biogeochemistry. My sketch names some of these parameters like currents, water temperature, and salinity, wind speed, solar radiation, and nutrients. We name all these parameters covariates because water quality varies temporarily with these parameters in a certain manner and in the short or in the longer term. Time series forecasts of some of these covariates will be available through daily marine and weather forecasts. Daily geochemical forecasts may not be available or may be available, depending on the site you consider. Let us just be flexible here on what sort of forecast is available. Last but not least, there are parameters which do not vary much temporally, but do vary spatially, like symmetry. These parameters do not vary temporally with our water quality parameters, but may vary spatially with them. Altogether, inferring future values of water quality parameters from their past values and from past and future values of their covariates is our time series forecasting problem. Note here that I have not illustrated the spatial dimension of this problem. To account for lateral spatiality, we consider the values of water quality parameters for pixels next to a target pixel as another sort of covariates. Why do we want to forecast water quality by means of spaceborne observations? Spaceborne maps of water quality parameters like chlorophyll concentration and suspended matter concentration are provided daily at medium to high spatial resolution by, for example, the Copernicus Marine Service. The Copernicus service also provides daily forecasts of water quality parameters at coarse spatial resolution based on biogeochemical models, which are fed from various data sources. These forecasts are model-driven and thus are also limited by the capabilities of the modeling. Daily forecasts of water quality parameters at higher spatial resolution than provided by the models are of high interest to mitigate regional risk for public health, environment, and industry. And state-of-the-art deep learning methods could have the potential to achieve forecasts of water quality at these higher spatial resolutions based on a mostly spaceborne data-driven approach. After now having explained the problem, the further outline of my talk is the following. I sketch our study plan. I explain the study sites we have selected. We will consider coastal sites as well as inland water sites. The definition of sites in terms of their geographic boundary is yet somewhat provisional. I present an overview of parameters and their sources for our coastal sites. And I compare some candidate machine learning methods we have found in the literature in terms of their capabilities and applicability to our problem. I briefly sketch our training and validation method and conclude my presentation with the study profile sheet. So the first cornerstone in our study plan is the provision of data cubes for the selected coastal and inland water sites. We think that these data cubes will have some value outside of the context of this study too. For each site, we will train a machine learning model which shall facilitate credible daily forecasts of turbidity, chlorophyll concentration, and occurrence of algal blooms up to several days ahead. The forecast shall include an estimate of uncertainty, like prediction intervals. Each model will be validated at its primary site, but we will also test the applicability of the models to other secondary sites. An overarching activity which within our study is the exchange with the scientific community, which is why I am giving this talk. And we will conclude the study with a future roadmap and possibly a scientific publication. We selected two primary coastal study sites, the German Bight and the Belgian and Dutch coast. Both sites are well known to us and our partners through activities within the context of the Oslo-Paris Convention. Availability of observational and model data and in situ data for these areas is excellent. We also consider a single primary inland water site. It will either be Lake Mälaren in Sweden, Lake Trasimeno in Italy, or the Alster, which is a dammed up river here in our home city, Hamburg. Again, all sites are well known to us and our partners from many years of work on water quality projects. We selected the Gulf of Finland in the Baltic Sea as a secondary coastal study site, which we will use to test the transferability of our coastal models. The Gulf of Finland is also well known to us and our partners through activities within the context of the Helsinki Convention. We will also select a secondary inland water site, but we have not made the final decision yet. Anyway, it will either be Lake Mälaren, Lake Trasimeno, or the Alster. We will collect data from several sources to generate the data cube for each site. For all data, we consider a period of 5 years from 2018 to 2022. Chlorophyll concentration and turbidity or suspended matter concentration will originate from daily level 3 Olchi water quality products, which are provided by OSPAR or the Copernicus Marine Service. The availability of the OSPAR product for this study is not clarified yet, but OSPAR is our preferred source of data. If this is not available, we could use Copernicus. The data have a spatial resolution of 300 meters. Daily reanalysis data on physical ocean water parameters like mixed layer depth, salinity, temperature, and currents are provided by the Copernicus Marine Service at a spatial resolution of 7 kilometers. Daily reanalysis data on physical ocean water parameters, on geochemical ocean parameters like nutrients, dissolved oxygen, and acidity are provided by the Copernicus Marine Service as well, also at a resolution of 7 kilometers. Daily reanalysis data on atmosphere parameters like wind speed, precipitation, cloud cover, and surface shortwave solar downward radiation are included with the ERA 5 dataset provided by ECMWF. The data have a spatial resolution of about 30 kilometers. Finally, static data like bathymetry and mean dynamic topography are provided by the Copernicus service and are included with their physical and biogeochemical products as attachments, so to say. We have used data science web channels to find suitable machine learning models, which are particularly useful for time series forecasting. Our most promising candidate is the temporal fusion transformer, TFT. Another candidate are gradient boosting decision trees. And yeah, we have some experience with using the classic multi-layer perceptron neural network for retrieving water quality parameters from space observations. You may know our C2RCC processor, which includes such a neural network. The table here lists the multi-layer perceptron for reference. The temporal fusion transformer is a multi-horizon deep learning time series forecasting model. The other methods are general purpose methods, The temporal fusion transformer and gradient boosting trees are considered state of the art. The MLP is not state of the art anymore. The TFT is readily applicable to the structure of our time series forecasting problem. It supports past to present observations, future forecasts, and static variables. It has a notion of time by design. The other methods do not have a notion of time and would require specific tailoring to take advantage of the knowledge of the problem structure. The TFT is capable of forecasting multiple time steps at once. The other methods can in principle produce output in any form, thus they could also produce multiple time steps at once, but it may be more appropriate to conduct recursive one step ahead prediction for these models. The TFT and the gradient boosting trees provide an estimate of uncertainty of their forecast, which is essential for us. And yeah, you know that machine learning methods are often criticized because of their learned features are difficult to explain and comprehend. Thus, often they are like a black box. But both the temporal fusion transformer and the gradient boosting trees do provide some insight on their learning on their learned features, which helps to mitigate this black box-like behavior of machine learning methods. Gradient boosting trees are the winner of the most recent Makridakis time series forecasting competition. The TFT did not take part in this competition, but outperformed some statistical and some other deep learning models in other comparisons. Documentation in code exists, fortunately, for all methods. For each side, we will consider the 4 years from 2018 to 2021 for training the machine learning model. The year 2022 is reserved for validation. We will evaluate the accuracy of our water quality forecasts with respect to actual retrievals of water quality from space observations. And we will compare our water quality forecasts to in situ measurements too, where and when available. Due to representation errors, in situ measurements, however, will probably be quite different from the forecasts, which are based on space observation. Yeah, I conclude my talk with the study profile sheet, giving you a summary. Thank you very much for your attention.
Wonderful. Thank you so much, Ralph. If we have any questions for Ralph, you may unmute at this time. You can raise your hand. You can put your questions in the chat. Can you let us know, just remind us, I'm sure you said it in your talk, but I might have missed, what's the time horizon for this project?
The time horizon is 1 year. So we started in
Oh,
September and end will be August next year, maybe
okay, so
mid-September because we started in mid-September.
I do see it on the screen, but I thought this was like the first part of a longer project. I'm so sorry.
No.
Okay, wonderful. Any other questions for Ralph? Okay, I'm seeing Rick says, you noted that some machine learning methods have explainability. This is important. Can you provide references
Yes, I will
for?
do. So yeah, I think hyperlinks are included with my presentation. I think yeah, I will add them to my presentation on an additional
Yeah,
slide afterwards,
or
okay, and send
or
this to you.
if they are accessible and you can just pop them in the chat, I think that would
Okay,
be
yeah,
helpful.
okay.
I will certainly add them when it's posted on our website. I will certainly add the links
I will do both.
as well.
If
Yeah.
otherwise, in the chat, things get sometimes lost.
Okay. Any other questions? Well, if there are none, and if our presenters for the next talk are ready, we can move
There
on.
is there is one more question in the
Oh,
chat
I'm
from
so
Felipe.
sorry. Felipe says, thank you for your talk, Ralph. Is the model that you are training, validating, would that be available for other end users?
In fact, I'm not sure about that. I mean, this is an ESA project, and I'm not fully aware of all the contractual conditions. But the aim, at least, is that we have some public activities here in the project. So the idea is that we will publish our data cubes. Not sure if the end models will be public, but from our side, from my point of view, we could make them public. But, I mean, Carsten has the last word, and the final word really has the contract.
Yeah, usually we do our things open source, as you know, and I don't think that there are any constraints from our funding agency on this.
Okay, it's great to have the head honcho on the call to make the final decision. Irene writes, Irene Politi, do you expect that 4 years of training data will sufficiently capture those unpredictable climate extreme events that may affect the forecasting model's capabilities?
We hope. I mean, climate events, we do only short-term forecasts up to several days, so I'm not sure whether climate events is the right word for this. But at least we hope that our databases will be sufficient for doing these short-term forecasts.
Certainly have had several anomalous years lately there in Europe. So fingers crossed that it works. Okay, any other
Yeah.
questions? Either unmute or ask your question in the chat. Okay, if you have still have questions or are struggling to find your button, you can go ahead and type it in the chat, and Ralph and all of our speakers will be able to peek in the chat and perhaps answer the question there in the chat. One more round of applause for Ralph for his presentation, the preliminary
Thank you.
forecasting from space. And if our next presenters are ready, I'm going to transfer control over to them. So I find them. Here you are, Abangalos. Make presenter. All right. Is that working for you, Evangelos?
Yes, yes, I think it's working. Um,
We're seeing your screen, but not your
yeah,
presentation.
now,
Yeah.
okay, you
Perfect.
can see my screen. Okay, okay,
Yes.
thank you. Hello from Greece. My name is Vangelis Romas. I'm head of the ENVIS Research and Development Unit. And today, I would like to give a short presentation about some machine learning algorithms that we have developed for forecasting algae dynamics in freshwater reservoirs. So the work I'm going to present has been part of the Prime Water project. Uh, Prime Water was a 3.5 year long research project funded by EU that concluded a few months ago. And its main aim was to combine EO-based water quality products with hydroecological models in order to generate operational short-term forecasts and identify at an early stage changes related with water quality issues such as algae blooms, so that downstream water management and end user can use this forecast and improve their decision making by proacting instead of reacting, and thus operate more efficiently. So the project was coordinated by us, by ENVIS, and had an international consortium with partners from Europe, United States, and Australia. The same applies for our case studies, where the various concepts and techniques of prime water were tested and demonstrated. We had in total 5 case studies, 2 reservoirs in Sardinia, in Italy, one reservoir in Ohio, United States, and two more in Australia. So for each one of these case studies, a different combination of services and functionalities were deployed, including EO-based water quality monitoring, short-term hydrological forecasting in the upstream catchments of the reservoir, water quality forecasting inside the reservoir, both with process-based models and machine learning solutions, early warning systems, and even more customized solution, for example, for the optimization of interconnected reservoirs, which was the case for the Sardinian case study. So, in Prime Water, ENVIS was in charge for the water quality modeling, and one of our primary goals was to examine how all this wealth of multispectral satellite data can be used for short-term hydroecological forecasting. And this was achieved in 3 different ways. The first one was to evaluate the increase in performance of three-dimensional process-based model when Earth observation are assimilated into them using data simulation techniques. The 3D, the process-based models, we are using the Delft suite. It is a coupled hydrodynamic model which resolves the circulation pattern inside the reservoir across different layers. And then we are running from the same suite a water quality model which describes algae growth as this is limited by nutrients and temperature, radiation, etc. The second one was to examine if we can leverage satellite data for developing credible and explainable machine learning models for prediction of phytoplankton dynamics. And the last one was a hybrid solution where we coupled process-based models with a mechanism for correcting errors based on data-driven solution. So today's presentations focuses on the second item, on the machine learning models, and specifically the models that we developed for predicting chlorophyll A variation in one of our case studies in Lake Hume. So we followed a theory-guided model architecture by evaluating the relevant importance of some critical hydrological and meteorological factors that we'll already know that affecting algae dynamics, like nutrient effluxes from upstream catchments, radiation, and temperature. And we adopted a sliding window prediction strategy, which assumes that the current chlorophyll A conditions in the reservoir are a function of what have been the hydrological conditions in the past X days and what have been the meteorological conditions in the past Y days. So for Lake Hume, the optimal length of those sliding windows were found to be 15 days long, both for both families of predictor variables. However, for other case studies, we have found that differentiating those windows can lead to an improvement of performance. So meteorological and hydrological conditions are used as input, and the machine learning models try to predict chlorophyll A concentrations. So for Lake Hume, meteorological data were obtained by ERA5 land dataset for a 5-year-long period, 2015 to 2019. And hydrological data were obtained through the Australian version of HYPE model. So HYPE is an open source hydrological model that was set up and calibrated by SMHI, which was partner of Prime Water. And the model was set up in all project case studies and provided operational 10 days forecast at catchment level for river discharges, water temperature, but also for nutrient effluxes and sediment effluxes entering the reservoirs. And this was a very valuable input for the water quality models. So for the training of the machine learning model, we used the chlorophyll values obtained from Landsat and Sentinel multispectral data. And these were processed with EOMAP semi-IP algorithm. And in total, we had almost 150 images available. We used two distinct machine learning families, random forest and Gaussian process regression. Random forests are, let's say, a human readable model. It is easy to interpret most of the times. It is robust and handles input noise quite well. And they can also predict either continuous variable or class labels. So they are also suitable for classification problems when, for example, you are not interested in the exact value of a variable, but you simply want to know if your variable, so if chlorophyll A, for example, will be below or above a certain threshold. On the other hand, the Gaussian process regression is based on a probabilistic technique and usually produces models that are not so easy to explain. They are more like a black box approach. Their strong point is that they can provide uncertainty estimations of the predicted variables. And the random forest and GPR models were deployed in 4 different areas of interest in Lake Hume. So in total, we had to set up and train 8 different models. Now, the following graphs present the result of the machine learning models. For each area of interest, we have with blue dots the chlorophyll A observations update from satellite imagery. And with the 2 continuous lines, we have the chlorophyll A values predicted by the machine learning models, taking into consideration only meteorological and hydrological conditions of the past 2 weeks, which was found to be the optimal sliding window for Lake Hume. So in those graphs, we are seeing that both machine learning models perform well in all areas of interest, and they are capturing satisfactorily the temporal variability of chlorophyll A. The mean absolute errors were similar for both models, ranging from 5.9 to 9.7 micrograms per liter. However, the GPR model offered a smoother prediction. This is a common trait of GPR models to smoothing out the results. Now, regarding the timing of peak values, we discovered that this is accurately captured. However, there is a consistent, let's say, tendency to underpredict high values of chlorophyll above 30 micrograms per liter. And this is a common problem with when using imbalanced datasets. And so while we had almost 150 observation points, the peak values were a very small percentage of them. So the model tends to treat those values as noise and ignore them. So in general, with ML, it's more difficult to predict events that happen relatively rare. Therefore, the chlorophyll A extremes were captured correct in terms of timing, but they were slightly underpredicted in terms of magnitude. Now, regarding the model interpretability and the importance of individual meteorological and hydrological predictors in the models, we have made an analysis to identify which were the most critical ones. We noticed that there were not significant difference between the GP and RF models. However, we did notice differences between the areas of interest. So what is important in the north part of the lake might not be equally important for the south part of the lake. But as a rule of thumb for Lake Hume case study, we can say that the contribution of nutrient influxes is what mostly drives phytoplankton dynamics. However, in other case studies, radiation or temperature have been found to be dominant in this process. So after having evaluated, let's say, the predictive capabilities of the machine learning models and felt confident about their performance in heat gas mode, we wanted to examine how these models perform and behave in forecasting mode. So we set up a 4-year-long reforecasting experiment where we just fed the models with expired forecast of meteorological and hydrological drivers without training them again. So to benchmark this performance, we compared the ML models against a naive alternative, which is the last known observation. So this implies that in absence of information, the chlorophyll values in the future days will be the same as the most recent observation. This is also known as the persistent method. So if the machine learning models perform better than this persistent approach, this means that they do carry added value not only for predicting current conditions, but also for short-term forecasting. The comparison was performed using the mean absolute scaled error. In this indicator, lower values denote a better performance, so 0 is the perfect model. And by definition, the persistent approach has an error of 1. So if the scaled error of the machine learning models are below 1, this means that they are outperform the naive alternative. So in these graphs, we are seeing that for the 4 areas of interest, how this scaled error varies as we are increasing the forecasting horizon. As we can see in those graphs, it's clear that the RF models were superior to the GPR alternatives for all areas of interest and for all lead times. So you can see that the dark blue line is always below the light blue line, so their errors were lower. And now by comparing the RF with the dust straight line, which is our performance threshold, we also see that the random forests were better than their naive alternatives for up to 10 days. Of course, as it is expected, when the forecasting horizon increases, performance slightly deteriorates. But this still happens within acceptable limits, as you can see in the at least in the first 3 graphs, where even for 10 days ahead forecasting horizon, the RF are better or equal to the native alternatives. In the 4th case study, you can see that random forests are almost from the first day of the forecasting horizon comparable to the naive alternative. And this happened because the 4th area of interest was a data scarce area, so we didn't have as many data points as the other 3 ones. And another interesting conclusion from our analysis was that the performance drop, this performance drop that you can see as the forecasting horizon is increased, is attributed mostly to the uncertainty of the meteorological forecast rather than the uncertainty in the hydrological forecast. Yeah, this is my final slide that summarizes some lessons learned from all prime water case studies. So in prime water, we have demonstrated that multispectral satellite data, apart from being used for data simulation in process-based models, there can be on their own a valuable source of observations for data-driven applications. The random forests were found to be consistently better than Gaussian process regression, at least at the case studies examined in prime water. And regarding forecasting, we have proved that it is feasible to develop credible data-driven models for forecasting phytoplankton dynamics, which are better than naive alternatives for up to 10 days ahead in time. And finally, I would like to stress once again the importance of data availability in creating some credible machine learning models, which it is not only a matter of absolute numbers, but also of data representability, since a rich in points dataset, which, however, does not have enough observation of peak extremes, might still be problematic for machine learning applications. So these are all the contributors and partners of Prime Water project. You can also visit the site and the operational platform. And thank you for listening.
Wonderful talk, Vigilis. I do have a pocket question, but I will defer to the audience first. If anybody has any questions, you can unmute yourself. You could type it in the chat, and I will moderate it for you. So while folks are trying to find their unmute button, I'll go ahead and ask you my pocket question, and it also goes out to Apostolos or any of the other speakers today. I wonder if you, anybody could comment on what you think the biggest hurdle facing the water quality forecasting community is today. Aside from the obvious, of course, resources to sustain a forecasting effort, I guess. Is there one thing that the community should tackle first before anything else? The silence makes me think that that's a loaded question, maybe, that I'm asking. Apostolos, please unmute and listen.
Hi, Mary Beth. Hi, all.
Yes.
From my side, I mean that's a very interesting, point you're raising. Well, we it's and I think the answer is should be seen in multiple angles beyond the technological or the scientific ones, because from that point of view, obviously there is a very good space for new ideas, for new techniques, for new concepts that can give us gradually more and more skillful, let's say, data and services for water quality forecasting. But this is one thing. But at the same time, um, I feel that we cannot actually detach this, let's say, supply side from the demand side, and I think also there is a very the challenges, significant challenges also exist there on how to use those new, let's say, predictions or predictive capabilities, how to maximize their adding value, how to make them useful, how to, and whether we can include them, incorporate them in actual downstream services and users' needs. So I think it's not easy to just describe the challenges or the opportunities in just on technological terms when it comes to forecasting services, and we saw that clearly in our project in Prime Water. I'm not sure if if you were expecting this kind of response, but
No, you're good. Thank you for the clarity, and I certainly welcome anybody else who would like to comment on that or ask another question. I will tell you that so there's quite a lot of interest in delivering a forecasting service at the GEO Secretariat level. Um, uh, and it's not just water quality forecasting. They want to do, a variety of forecasting. So the interest is there, even if maybe the science isn't there or the science isn't optimized. And I just wondered if there were other challenges besides the science that maybe we needed to also consider. Thank you. Any other questions for Vagelis or Apostolos on their talk or any of the other speakers? This is not the first time that I have seen the outcome of the Prime Water, and every time I do, I'm struck by something new. I find something new in the results to focus on. So certainly, I think this series of second in the series of forecasting talks, as well as our upcoming webinar on November 7th by Mar Rakamora, will be useful to set the stage for our biennial meeting discussion. That will happen on November 13th, where we as a community will sort of gather some additional input, as well as these recorded State of the Science webinars, and just have a round the table discussion on the feasibility of forecasting for water quality, and what we should tackle to try to make that happen. So I definitely thank all of the presenters. Are there any folks in the audience who would like to ask a question about the Prime Water project outputs? I do think your product is now completed, and that your final report is out. Is that correct, Vagelis and Apostolos? Your final recommendations for Prime Water are public?
Yes, the project has completed on early June, and all the deliverables are available in the Cordis. Also, the platform, the operational platform, is you can visit it on the link, and in the website of the project, there is also a virtual lab. This is a repository where we have uploaded almost all of our data set that were used either in the process-based models, in the hydrological models, in the machine learning models. So everybody is welcome to download those data sets and experiment with their own algorithms, perhaps, and do a comparison. This is all what those scientific projects are about, comparing results and finding, let's say, new opportunities for collaborations.
Yes, thank you. We have enjoyed presentations on the virtual lab by Claudia Giardino and others in the past, and it is quite useful, and there's a link to it on our website as well. So yes, that is a useful tool. Well, I just want to, on behalf of everybody at in the management team and the steering committee of GEO AquaWatch, extend a extra special thanks to those who were able to present today and in the past, and also our future forecasting webinars, because we did extend a special call to specifically speak about the potential for forecasting as opposed to some of the other water quality work that the community is used to presenting on these GEO AquaWatch webinars. So we do especially appreciate everybody who contributed to the forecasting state of the science. And we absolutely thank you for joining us today. Today's recording will be on our website probably in a couple hours. And of course, we wanted to also point out that we do have the early career scientists water talks. We had our first one yesterday. We have another one coming up on November 29th, as well as January 9th. We do have 4 in that series. It's an excellent way to find out about new science, meet early career scientists, learn novel techniques, as well as help us amplify the voices and science of scientists from the global south, which is the goal. So thank you for joining us today and have a wonderful day. Goodbye. Oh, Philip, did you want to ask a question? Sorry.
Bye bye.
Bye bye. Thank you.